So today we're going to be learning the steps of the scientific method. In your binder, this is going to be under notes, and it's going to be Unit 1, page 4. I would title your notes Scientific Method. So the scientific method is going to, in general, be a list of steps scientists use to solve problems. In the figure, you'll see a lot of these steps, but what you'll also notice is there's not a linear direction of this method. You'll see that sometimes the scientists will get stuck in where their experiments are going, and they'll have to restart. And this is normal. I also want you to note that scientists really have to communicate their results, and that's something that we often forget that scientists have to do. All right. We'll go through this process together. So I would label number 1, and then in a big title on your note sheet, say state the problem. So under state the problem, you want to write some bullets, but make sure you're saving yourself some time during these notes by abbreviating. So when a scientist states the problem, they're going to first make some observations. They don't make up a problem from nothing. They have to have seen it. And they're usually going to write the problem in the form of a question. Under another title, you'll write background research. Scientists will collect information on that problem that they've just stated. They'll usually do that and become an expert as much as they possibly can. And they'll sometimes go to the library, but often these days, most of this research is going to happen online. And for our class, you're going to be doing a lot of research with the librarians about our plant mutants this year. And that's going to be something we learn a lot about. How does background research really work? All right, not until after those two things does a scientist state a hypothesis. Now, a hypothesis isn't just a guess. A hypothesis is an educated answer, not a guess. And it's an answer to a problem. It's educated because, well, they just were researching the problem. And so they now are making some sort of answer based on what they found out. And usually you guys have known that in the past you've written this as an if-then statement. Not all scientists always write it this way, but we're going to do that because it's best practice. Alrighty. Then after that, they've set up the experiment. And in setting up the experiment, they have to figure out a procedure. And under that procedure, they're going to have to think about what materials they need. They're going to have to think about what kind of safety precautions they'll have to take and all that sort of things. So here's some of the really big important vocabulary that we've been learning in class. And this is a review. So an independent variable is the variable that is changed by the experimenter. And we're going to remember that, and we talked about this little saying in class, that I change the IV, right? And that's going to be I as the experimenter and 1 changing the independent variable. And when we think about graphing, the independent variable is always going to be plotted on the x-axis. That's the bottom horizontal axis. We have another saying to remember where the x-axis is and how it relates to the independent variable, and that saying is X-Men fight for independence. Now, do you have to memorize these sayings? No, but they'll probably help you remember what the definition is and where the independent variable goes on the graph, and that's super important for our class. Secondly, there's the dependent variable, right? The dependent variable will depend on the independent variable. But that's a little confusing to remember. So what I'm going to recommend you thinking about is that the dv, the dependent variable, is going to be plotted on the y-axis. But I like to remember that d and dv really stands for data. It's the data the experimenter is going to collect. And I know that's not stated on this PowerPoint, but that doesn't mean you shouldn't write down what I just said. I really think you should write down the dv is the data. Next, this is one of the things that students always get confused on, a control group. A control group is the group that's unchanged. It's used for comparison, and it's going to help us figure out what's going to be the background information, what's the normal conditions. Oftentimes, control group is going to be water, and not just water, distilled water. Other times, it'll be plant that's just normal. And in our Arabidopsis project, the normal plant is called a wild type plant. Now, often students get this confused. this control group confused with constants. They sound the same, but they're totally different. Constants are things that stay the same. Constants in experiments make them fair. If I'm trying to change too many things at once, I can't really tell cause and effect. So some examples in many experiments of constants are temperature and sunlight. If I want to see the impact of how much water I'm watering a plant will have on its growth, if I'm also changing temperature and sunlight, I don't really know if my effect is due to the temperature and sunlight or the water, which is really what I'm trying to look at. Repeated trials is going to allow us to figure out how accurate and how reliable our data is. Usually in science, we want to have at least 3 trials in order to find our data reliable. And this is going to be in case we have mistakes or errors, and those will be then reduced. Now that we've really set up our procedure really well in our experimental design, we can collect data and results. And when we collect those data and results, we're going to make sure we observe the different types of data in our experiment. Now, we've talked about in class that there's 2 main types of data. Quantitative data, which you'll see the N in quantitative will help you remember that quantitative data is numbers. And then there's qualitative data, which is words. An example of quantitative would be 40 centimeters. An example of qualitative data would be the rose is red. What would you say about the quantitative data on a frog? We could say that there's 4 legs because that's a number, that's quantitative. I could say the frog is green, and that's qualitative. Lastly, we're going to write a conclusion when we're finished with our experiments. We want to think about what was the purpose of this experiment in the first place, and that might take a while to remember and revisit our background information and our problem. We're also going to revisit our hypothesis. What did we think would happen? And then once we're looking at our results, well, what did the data show us? And lastly, when we're going to conclude, we're going to think about, okay, I thought this would happen, but the results show this, so therefore the data are telling me that in reality this is what happened. And I might want to also say, but maybe I didn't have enough trials, and that might have caused some error. We'll talk about this process a lot this year, especially with our Arabidopsis project. And lastly, we have to have some reasoning. I'm not going to ever have a conclusion that's not justified with evidence. And often those evidence, this why or why not, can be backed up with the data, graphs, tables. And that's it, guys. Good job. You made it through our first podcast. See you later.